Welcome students. We are again continuing this application of various chromatographic techniques in herbal drug standardization and herbal formulation and drug standardization, and in which we are last class we just discussed about the TLC, and then now we are going to have discussion about the high performance thin layer chromatography. In high performance thin layer chromatography we will come across this analog of digital converter, the name that is ADC in some cases, so that's why I mentioned that. You see this high performance thin layer chromatography, just let me pull it down. High performance thin layer chromatography is a powerful analytical technique which is wonderfully suitable for qualitative and quantitative analytical task. The development of chromatographic fingerprints plays an important role in the quality control of complex herbal medicine. And here there is a video which I can give you some idea. Well, already I have shown this in another topic specifically in herbal drug technology. Here we can see that is about the fingerprint for ginkgo biloba, and the sample preparation of analysis and sample application, and we can see there is one more. I will just tell you, this is the list is needed for you to know about the sample preparation specifically. Here you see here sample preparation of analyze, that means analytical product, analytical substance which you want to prepare, sample application and development, documentation, derivatization and documentation. Again we used to do. This is the steps which she is doing here. So once you know about these steps, it will be easy for you to understanding. Here you see you have to prepare reference sample, and also you have to weigh 100 milligram of sample and reference, and you can addition of 10 ml of methanol. You have to prepare the sample preparation. That's what we are mentioning here, and so that you can see how she is. You just see she is weighing of milligram by means of a chemical balance, digital balance, and then she is adding methanol and preparing the sample for the HPTLC, for preparing the exactly she is preparing the sample and keeping ready. And now she is going for you see here the sample preparation. You need sonification for 10 minutes, centrifugation for 5 minutes in 5000 RPM, and transferring samples to vials. So these three things will be happening now in the video. You can watch that very clearly. Sonification means not by means of using stirrer or anything, by ultrasonic waves you will get sonification, mixing of the solvent system, methanol which you have added in the test tube. It will mix it clearly, and then after mixing you are giving centrifugation in 5000 RPM speed, and then you are transferring the sample in vials, and it will be sealed as shown. She will be showing now. So you can see now this is the place where she is keeping for sonification, and ultrasonic waves will move through the test tubes what she has given methanol, and then she is keeping it for swirling. There is one instrument where then she is keeping swirling, is that small one blue color instrument, swirling, and then she is keeping for centrifugation for 5000 RPM. It will be given, and then after that you can see this. You see how after centrifugation supernatant liquid is taken by means of micropipette, and then micropipette, and then it will be transferred to the vial, and it will be sealed by the, very interesting to see how she is sealing the sample for standard, everything you have to label properly, and you have to keep, because every day you will be doing different, many samples will be done. So she is just pouring it into the vial and sealing with aluminium cap and keeping ready for sampling to the instrument, for HPTLC instrument. So you see now she is coming, and now what all the things you have to prepare. Preparation for the analysis, that is loading and excluding vision, that is cat is the, there is an it is slightly hiding, that means it is the method which they are following. Filling the sequence of the table in the computer, all the names, what is the what is the solvent system which you are using, what is the herbal drug which you are going to use for HPTLC, labeling the HPTLC plate by means of a pencil, and capturing the image in the computer by means of a normal light and UV light in 254 nanometer and in 366 nanometer, and before derivatization the place can be visualized and be taken, photographed, can be recorded, and the spots can be identified. So this is capturing of the image and cleaning the plate under white light, re-emission, transmission and UV in 254 nanometer, and 360 also can be done after in the derivatization place. So you see here now I think she is after sampling, as we had after this mixing. After this one we have come up to here. Now she has taken the sample, and this also we have watched. After the sampling, she is now sealing it by the sealing, and now we are just coming to this part which we are discussing now, and how they are computers. In computer you see there she is recording about the this ginkgo biloba is the sample, and what is the solvent system which is using and installing it, and then she is giving all the details, what is the date and what is the sample size and how much ml which she has she's added to the sampling vial. These all things should be recorded clearly in the computer, and it should be fed for the sampling area. Now if you see, this is the instrument that is she was feeding is very important, documentation and the sampling feeding, and ginkgo biloba, how many samples, how many spots you should be mentioned, all these things she will be detailedly given. And once she is giving label to the plate, you see there is a ready-made plate, she is writing the date and writing the name of the researcher or the worker and the sample and the date time, and she is keeping now to visualize the plate. First she has to visualize the plate by keeping this in the chamber, and she is coming to visualizing in the light, that is normal light. You see normal light, the plate looks very clearly like this, and the sample application now. Afterwards she is going for the sample application. Sampling, you have to keep the plate, you can see the plate is kept in the video, it is kept in the place where the sampling area, and she is just in the slot and just closing the slot, and so that you can see you have to click in the computer, give the command for the sampling. So when she gives the command, immediately the sampling spot, you see how minutely it is keeping in the plate, very clearly it keeps a band of spotting, very minute micro spotting can be kept because you have kept already. After spotting you have to rinse. This rinsing solution, you see rinsing have to be done by means of methanol, and you have to just rinsing, and then after rinsing the this is the chamber of the HPTLC, and you can see by means of rinsing. After rinsing, what is the thing? What are the things? Humidity, the development of the digital or the development of the ADC, the humidity control 33 degrees and reproducible. Filter paper is back will be there for saturation you have to keep, and 10 ml is for developing solvent, and 25 ml is for developing solvent saturation. For example, you have to go for saturation. So there is two tubes. There is two provisions will be there in the instrument. One place it is for in the base, there will be 10 ml. Another place for saturation in the chamber completely we are using 25 ml. So you can see here now they just start doing. She is taking for the chamber. She is keeping for saturation also. She has kept filter paper behind, and you can see the plate is also kept. Sorry. It is fixed. Now she is taking for 25 ml of solvent system, which is kept there for saturation. This is for 10. This is for 10 ml for the developing, and again she is taking for saturation separate. This side there is one provision for adding for saturation. You can see for saturation you have to add here 25 ml should be added. And then after adding you can keep the plate. Now you can keep the plate in the slot, which is already your sampling you have done by means of automatic sampling, and you have to keep the slot which is already labeled and fix it the plate there, and then the plate, and give the command for developing. So when you give the command, the plate will come down and it will spark one light and started the high performance liquid chromatography or a thin layer chromatography. The moving, the plates are started, the light is blinking. Once it is coming to the area where it stops, that is 3/4 of the plate, the solvent system moves, it will stop, and then it will stop, and then you have to take the plate. Now you have to remove the plate, because the plate, the development is over, and then you have to take the plate and now go for drying and visualizing. As I told you, you have to visualize white light, and in UV 250 nanometer and even 365 and 66 nanometer. The plates will be visible, and you can see the plate very clearly. The picturizing can be done. So there you are keeping there, and she will close. This is the chamber which can be visualized. First white light, she is visualizing in the white light of the white light. She can see that this is by white light. There will be some spots lightly, but it may not be seen very clearly. And now she is going for only when not clear. You can see some spots are there, but this is for 254 nanometer when you go for visualizing, and this is for 366 nanometer higher UV light. When you are seeing, you are getting the spot very clearly. Now you can go for derivatization. Derivatization means, wait a minute. Derivatization means, you can see here, this is the plate developed, and now the next step we are going for derivatization. So what should be done for derivatization is, what I want to say here is, derivatization means heating the plate for 100 degrees centigrade 3 minutes and dipping in a natural product reagent. There is some reagents are there which will cover this. In the class also I told you how will you make the chymograph which you prepare in pharmacology. We will go for colophony, or we will give for some permanent, the graph should fix it. So like that, this can be fixed with the reagent, and so that the color visibility is very clear for your RF values, and to identify the qualitative and quantitative estimations will be very clear for you to in HPLC can be performed. So heating the plate, this is called derivatization, and derivatization means heating the plate after development 100 degrees centigrade 3 minutes and dipping in natural product reagent and drying with a cool air and dipping in polyethylene glycol again for 400, and then drying and cool, and then you can see the color visibility very clearly. So that is what they are going to do. That's why I put in pass and to double. See, now the plate is given, brought, and she is drying it, and then she is going to after drying, and it will be taken to the natural. You can see the natural reagent for derivatization reagent, natural reagents. Even the name of the reagents also sometimes for specific drugs it will be given. In the natural reagent they are dipping it, and after dipping it again it will be dried. You can see the dryer can be used for drying it, and then polyethylene glycol. You see polyethylene glycols. Polyethylene glycols can be used for derivatization now, and then again you are drying it, and so that you are developing the compound. And so you see now your derivatization in white light, re-emission, and also you can go for finding out these different plates. You can just find out where the vision can be identified. So you see here now the plates can be identified by derivatization. You can see the light is very clearly in the white light, and then it is in 266 nanometer and 254 nanometer. You can see very clearly, and reproducible HPTLC plates can be identified very clear. And the second derivatization can also be done. You can do second color reagents can also be applied, and second derivatization also can be done. Like these plates can be very clearly seen. So this is what a very good interesting instrument which can be easily identified. So listen to this. You can write 100% sure, I am damn sure, HPTLC, how it can be applied for natural products and natural standardization, how it can be performed. By means of this video, it is possible for you to perform. And you see the chemical compounds, I have given some list also. For polar compounds like ethylene ethyl acetate and toluene, it is slightly slide because it was very big, I was unable to bring it down. So it is ethylene ethyl acetate and toluene, and as a solvent system which can be used for anthraquine glycoside, abertrain alkaloids, cardiac glycosides, bitter principles, flavonoids and saponins, and lipophilic compounds, essential oils, terpenes and coumarins, naphthoquinones, valproate alkaloids. These also for valproate, there are also toluene and for example ethanol and acetone can be used in certain ratios, and also alkaloids, toluene, ethyl acetate, diethylamine 70:20:10. Flavonoids you can use by means of acetate and formic acid, glycolic acid and water. 100:10:11:11:26 can also be performed for your various performing. And also you can, for saponin, chloroform, glycolic acid, methanol and water can be used in 64:32:12:8 ratio. And also for cardiac glycoside, ethyl acetate, methanol, water in 100:15:5:10 is also, or you can use in 81:12:10 also can be performed. And terpene, chloroform, methanol, water in the ratio of 65:25:4, and triterpenes, ethyl acetate, toluene, formic acid 50:50:15 can be used as a solvent system, and toluene and chloroform and ethanol also can be performed with 40:40:10, and also essential oil, toluene, ethyl acetate in 93:7 can also be performed, and ligands, chloroform, methanol, water can be used in a ratio of 70:30:4, and chloroform and methanol 90:10, and toluene and ethyl acetate 70:30 can also be used for ligands. And you can see for coloring reagents, for derivatization, as I told, there will be many natural reagents can be performed. For alkaloid, drag and drop, it forms a complex reactions and forms a nitrogen containing compounds. Should be orange red color can be seen, which is hidden in my picture. And natural compounds like polyethylene glycol reagents can be also used. Intense yellow, orange and green fluorescence are shown in UV of 366 nanometer can be seen for flavonoids. And diphenyl boric acid 2-aminoethyl ester forms a complex with 3-hydroxyflavone. When you are having a flavone, while condensation reaction, when you are adding polyethylene glycol, it gives a natural reagent that also gives a very good yellow orange color can be seen. Nimhydrin can be used for amino acids, peptides, amines and amino sugars, while yellow, brown and pink violet color usually can be seen in visible region. It's possible. And also iodine chamber can be used. Dark brown iodine spots can be seen for indole, quinoline, derivatives, thiols and all organic compounds. It produces iodine reaction, possible result in oxidative product, and 10% ethanolic sodium hydroxide and androquinone, amodine, reuen and androne, aloin Cascarosides can also be present. Which shows this is yellow color for cascarosides, and for androgen it gives reddish brown color can be seen. And then also you will see vanillin, sulfuric acid, and anisaldehyde, sulfuric acid, but bitter principles can be seen. Red, brown and yellow, brown, dark green zone can be seen in visible region, and also for saponins you can see color zones can be seen in visible region, and essential oil can be seen blue, brown or red zones can be seen in a visible region. So you can see now DNA fingerprinting, barcode like pattern generated by application of chromosomal DNA of an individual, which can distinguish the uniqueness of the individual from another, and also called DNA typing, genetic fingerprinting, DNA profiling and DNA typing. And DNA fingerprinting important tool in herbal drug standardization, usually for the identification of genuine drug from substituted or adulterated drug. And DNA finger genome can remain the same irrespective of the plant part used, while the phytochemical constituents will vary with a part of the plant, and the DNA sequence are highly specific, can be identified with molecular markers which can find out the particular sequence of DNA from a group of unknown. So this is also DNA printing, fingerprinting is also having a major role in the herbal standardization, and this DNA gel electrophoresis. So this is an electrophoresis method in DNA. The DNA molecules are separated on the basis of their charge and the size. So you have to remember the DNA are negatively charged. So as the concentration of the agarose, how you are preparing agarose increases the size of the pore, which increases the vice versa. So therefore the larger molecules cannot run faster in comparison with the small of the DNA fragments. So the smaller fragments appears near to the positive charged, migrate faster towards the positive node. Consequently, so once the electrical current passes through the gel, the DNA starts moving towards the opposite electrode, and a tagged fluorescent dye such as ethidium bromide used to visualize the DNA band. Another dye named DNA get loading dye monitoring the migration of the DNA by running ahead of it, and furthermore a known molecular weight DNA marker is used to calculate the exact DNA size. So you can see here that is ethidium bromide, or you can see homedium bromide chloride salt. Homedium chloride is an intercalculating agent commonly used in fluorescent tag, that is nucleic acid stain, and the molecular biologists laboratories for techniques such as agarose gel electrophoresis. So now we want to see that how they are performing this agarose and how they are performing this experiment. So you see here, especially we should know that there are many enzymes which is responsible for breaking down this DNA when you are giving a molecule of a herbal products, when you are giving, and there are 3,000, 4,000 enzymes nowadays available, and that enzymes will break down, and how much the breakdown of the molecule, like that the movement of the DNA will be there. So each DNA can be specifically, we can identify very clearly. So when you see this video, we will understand how they are preparing the agarose and making the gel, and very clearly. See here.
Restriction enzymes.
Let me give you how they are using the enzymes in the sample tube and adding with the enzymes, and so that breakdown of the DNA will take place as shown in the video. And now they are just after breaking down. You see, after breaking down this, she's adding the enzymes in the sampling area, sample point, and then after breaking down, we are keeping ready actually. And now we should know how to prepare this gel. The agarose you have to take, and in 70 degrees centigrade, the agarose, you can see agarose, now they are heating it for, it is in a 70 degrees centigrade, and it will be kept in the, and so that it will become liquid. and there will be a plate which contain, you can see there is a comb like things, that is a plastic, and the gel will be poured in a liquid state, and it is solidified. Now it is solidified. After solidifying, you take that plate, you will get all the wells. That is called the wells, where you have to keep the sample. And this is the nodes, that is the where there is two types of nodes will be there. Black one is negative and the other one is positive, and this is the electrophoresis, that is you have to kept inside, and the two electrodes will be, and also you have to take the sample according to the sample size also by the micro pipette. Here they are taking 10 micro ml is selected, 10 micro ml is selected, and also she is taking the samples, different samples. There are many samples she has prepared, and they want to find out which DNA is matching to this fingerprint, and they want to find out which quality, which one is related to which DNA. So that is why they are taking the samples, and by means of micro pipette, and then they are keeping in the well, that is which you have prepared. See, well means that the hole in the gel which is prepared, and see that by the staining fluid she is adding to the well exactly. It should not come split outside. Exactly in all wells should be different samples will be kept in there. And after filling all the, see, she has filled all with different samples. All the samples have been filled, and then she is closing the electrophoresis, and also she is putting the two electrodes, that is black and the red. The black is negative and the red is positive, and giving 100 ampere, 100 volts. 100 volts current is given, and it will be given for 30 minutes. So that what you can say, that is the DNA will be moves through the gel agarose, which is the plate. Inside that movement maybe towards the positive and the positive range, and that is very easy, and the technique which they have done. And now they will go for their visualization. Now that plates will be taken for visualization at the movements of the from the positive towards the positive, all the DNA will be moving, and then they take the plate and remove the plate, and you can see there is the plate, there is a movements can be seen, but when you are visualizing by computerizing, by the photographing, then you will see very clearly. Now you have to use that coloring reagent, that is ethenium bromide, which you have to just add as she is adding, and you are just making into color, and see in the white light she can see that very clearly. And so that you can see very clearly by this DNA where it is, you can see there is all the pictures which shows 1, 2, 3, 4, 5, 6, 7 samples she has given. In that which DNA is similar, you can see, observe very clearly. The second one and the fourth one is having a similar separation of DNA, very clear. So we can see these two are same and the other is not. That is, this is the culprit. So by this they will find out who is the DNA which is having matching together. So the sample number 2 and sample number 4 are showing the same DNA separation in the picture which you can see, observation which you can see from that particular the experiment they have done. So this shows that problem is solved very clearly. So very clearly it shows that they both are the sample. And you can see, even you can go for the very clear picture 2-3 times. If you want to perform, you can perform it and you can confirm this experiment very clearly. So this is how they will use this DNA fingerprinting by means of electrophoresis method, which is very commonly used, which is very common used for in herbal drug standardization. So that's why I have taken this also with along with the HPTLC. In the next class we will have a discussion about HPLC. This is also a very important topic which has to undergo, and HPLC is also plays a major role in all the pharmaceutical industry. So we should know about the instrumentation as well as the application of HPLC. We will have a discussion, thorough discussion about HPLC, and then we'll go. Thank you for being with me.